So I'm just going to kind of quickly go over a couple of our notebooks that focus on our two-photon calcium imaging. So to start, I'm going to go over, let's see. Okay, so this particular tool is called Suite2P, which is going to be gone over in much greater detail by the developer, Carson Stringer, on, I think, Friday. But this notebook it just has like a brief overview of using it. It has a really, really good GUI. So if you're actually trying to perform the segmentation that Suite2P provides, you could use that. But this is a demonstration of how to do it just purely in code. So essentially, Suite2P is used to segment the calcium imaging movies into actual regions of interest, which are, putative neurons in the field of view from the image. So that's why it's called segmentation. You're segmenting the image into regions of interest. And it does this using a classifier. You can use a number of different classifier models. This one uses a classifier that I trained you on the GUI. I think Carson will talk about this. But basically, we have a classifier model with the DataBook, and this notebook will load that classifier, and it will basically attempt to segment the calcium image in this NWB file and reproduce the image that the Allen Institute had produced from segmentation. So really quickly, it basically downloads the file here, and it runs Suite2P. This is basically just setting a number of input parameters for how the segmentation works. Like I said, Carson will talk more about this. And the output, you can see this is the fluorescence. Okay. So it comes up with a number of number of arrays basically in the output file. It basically produces a big directory of a bunch of different outputs of fluorescence metrics from all the different segmented cells, evaluations of whether or not that segment is an actual cell, and things like that, and then more statistics about it, which are here. But most importantly, what I want to show you is that this is the original segmentation from the field of view. And you can see it's very, very messy, but you could pick out specific neurons here, and you can filter it down to the ROI masks that it identifies that actually are cells. Cleans it up quite nicely. And so this is the one that was produced in the notebook, and this is the one that was produced by the Allen Institute during our pre-processing pipeline. And they look pretty similar, but not identical. So one note is that no two classifiers are going to be identical, and I don't even think two runs will be identical, but you can evaluate the performance using all those metrics that it provides and try to get good segmentation. So that's the first step in pre-processing our calcium imaging data. So you basically get the movie, and then you segment it into these cells, and then these cells have associated ROI traces, which I'm going to get into next. We want this. So, Saskia DeVries and two other researchers, she was collaborating with two other researchers in analyzing two-photon data, and what they demonstrated in their paper here, Mesa et al., was that this is kind of the analog of the ECU phys phenomenon I was speaking about earlier, which is that you can actually get pretty different results depending on how you filter down your cells. So this paper demonstrates that the different metrics that you're using to select your subpopulation of good quality recordings is very, very contingent on what quality metrics you're using and how you're selecting them. So there are, I think, 5 that are demonstrated in this notebook, and this is attempting to reproduce the findings that they presented in their paper. So this is doing some pre-processing here, selecting particular stimulus times, and plotting. Okay, so they presented 5 different inclusion criteria. Just for context, this is essentially what the event-aligned fluorescence looks like in this data set. And I mean, you can see pretty clear activity, pretty clear responses to particular stimulus events that were selected here. But how do you actually evaluate from looking at these event-aligned fluorescence responses if that unit could be considered actually responsive to the stimulus? And there's no agreed upon convention for this, and what you use has pretty big implications for your results. So I won't go over all of these, but like, for instance, this one picks based on just the mean or amplitudes of the mean evoked responses across trials, and you get this subset of cells. This one is uses z-scores, and you get a much, much narrower set of cells. This one uses a paired t-test and different set. And these sets are, I mean, they're not totally disjoint, right? But they actually have, the biggest thing that varies between them is how restrictive and inclusive they are. But the other thing is they do pick different, I guess, properties, latent properties in the subsets of cells that are selected. So I wanted to show you that because just to demonstrate that this is also an important phenomenon in calcium imaging. And then the other thing is that this notebook could actually be used to literally reproduce the findings in a paper really quickly. So you could, do this with a number of other things that rely on open data. So let's say you've segmented your calcium imaging video. You have used inclusion criteria to select cells. And what's another thing you might want to do? I know a couple of you mentioned that you wanted to look at chronic recordings. I think you guys are mostly looking at electrophysiology, but I think 2-photon might be more suited in a lot of cases for this. And the main issue with chronic recordings is that across multiple days, the cells move around. Image registration doesn't happen the same way every time. And so you want to make sure that you're actually looking at the same cells across multiple experimental sessions. So this notebook uses something called N-way matching, but there are other libraries and platforms that do the same thing. So this notebook demonstrates how you could do that if you're trying to look at specific cellular behavior across many days of chronic experimentation. And this runs it in like a sub-process here and produces a big set of file outputs. So this is the, I think, average projection image from the field of view. So it does some image registration, and it matches cells. Let's see. So to visualize the matching cells here, you can see it color codes them, and it doesn't have, this is actually a really difficult issue when cells are moving and changing shape. It doesn't have like an incredibly high performance, but in this particular case, you can see it matches quite a few of the more obvious cells here. So this is definitely something you can do for your guys' projects if this is, related to your question. And lastly, I wanted to talk about this notebook. I think yesterday Ben had mentioned the Zebra tool. And so this notebook, we partnered with the Mathis lab. We had an intern named Katrina Auger, and she made this. It's a really good notebook, and if you're interested in Zebra, that you could just leverage this for getting some really clear findings from the Allen Institute data sets. So the way Zebra works is it identifies lower dimensional embeddings in the activity. This is not exclusive to brain data. You could do any data set you're interested in, but identifies lower dimensional latent embeddings between populations of your input variable, and it's what's the word? It is unsupervised, and it specifically uses, let's see, it's described in here. Let's get through the pre-processing. Grid search. Okay, so yeah, it's unsupervised, and it basically is identifies the space through weighting the similarity of the activity of particular input cells. So this is done on our dendritic coupling data set. That and those other notebooks I showed you are as well. And that's going to be released, I think, in like the next week or so, which is a really rich data set. So I'd encourage you to look at that in a week's time. But I'm just going to show you here the latent embeddings. So this data set was designed so that we're showing them as a number of different natural movies and unnatural movies. And we also compare them to a phase-shifted version of the movie, which maintains the distribution of basically luminance and color, but it looks like nonsense to the mouse. So comparing embeddings across these phase-scrambled movies that repeat in sequence and real natural movies. And so it helps you identify actually, if the mouse is, computing a repeated pattern and actually learning too. So, for instance, this is the phase scrambled movie. The mouse, you can see a clear embedding over time in 3 dimensions. So you can tell that the mouse might be learning something or has some sense of the temporal structure of the video, but it doesn't look super clear. And you can compare this to the unscrambled version, which actually has it's just a video of mice doing things. And you can see we have clear embeddings. So that might indicate that the mouse is either paying attention more or just naturally tends to learn the structure of the movie more because it's something intuitive. There's also dots, which don't show really any structure in the embedding. And that's another phase scramble. Crickets show a pretty clear embedding. And so from this, you can actually predict, assuming you have the embedding and the content of the stimulus right, you can predict from the activity what the stimulus that was provided was. And this one was done relatively simply. It demonstrates how Zebra is pretty easy to use. This is doing a lot of optimization, but the actual line to use Zebra is just a handful of cells. It's after this. Yeah, this is essentially the training. So, Zebra is a really powerful model, so I'd encourage you guys to use it. And I think that is all I have for 2-photon. Any questions? Great. Good luck.